Hi, good morning or afternoon, everyone. My name is Sandro Matosevic. I'm an associate professor at Purdue University, and today I'll be telling you about how we're using natural killer cells as next-generation cancer therapeutics. I'll talk about how these cells are being formulated and engineered to be used in the treatment of advanced cancers. How we use NK cells in immunotherapy begins from understanding their ability at targeting and eliminating cancer. NK cells have an innate ability, they're part of the innate immune system, to destroy and attack and recognize pathogens, including cancer cells. And the killing, in its simplest form, is illustrated in this slide. NK cells have a lot of activating receptors and inhibitor receptors on their surface. If the signal points toward activation, they recognize a tumor cell based on HLA-MHC mismatch. They form an immunological synapse with the target cell that's been recognized. And then they trigger killing mechanisms. The killing mechanisms manifest themselves in the release of proteins such as granzyme and perforin, which pierce the membrane of the target cell and cause their destruction, or in other words, cancer death. This is a very accurate, precise, and effective process. And NK cells have high potency and ability to eliminate at eliminating targets. The mechanism by which they recognize pathogens allows NK cells to discriminate between a healthy cell and a disease cell so that they don't attack healthy tissues or healthy cells. So NK cells operate via multiple mechanisms. The most reported, studied, and common one is the missing self mechanism, which its basis recognition of targets based on MHC mismatch. NK cells, of course, are short-lived, so their killing ability and recognition of MHC deficient targets really lasts only for a very short period of time, 1 to maybe 2 weeks at the most. But because this mechanism is so precise, it allows NK cells to really operate in an innate way, meaning they're always on and they do not require antigen presentation. The diagram on the right side of the slide shows the different scenarios that NK cells encounter when they meet or when they're in the process of recognizing a target cell. So inhibitory and activating receptors, which are proteins on the surface of the NK cell, tell the cell whether to kill or spare the cell they recognize. If the balance is toward activation, the cell will kill, and what will happen is the target cell, which is illustrated in red in this diagram, will be killed. And if the balance of signals is toward inhibition, the target cell will be spared, which is shown in purple in this diagram. In cancer, we are hoping for, of course, a lot of red, or a lot of killing from the cancer cell, and really very little purple, meaning very little off-target or non-specific killing of non-target cells. These cells that are used in immunotherapy can be sourced from many sources. We mostly have so far developed NK cell immunotherapies from peripheral blood. These are oftentimes autologous, although the ability to use NK cell allogeneically, because of their ability to work under MHC mismatch conditions, makes them very attractive as allogeneic cell therapies. Cord blood is another popular source of NK cells for immunotherapy. There are more NK and T cells in cord blood that are in peripheral blood. Other two sources that are commonly used to source and obtain NK cells from are cell lines, and NK92 is a very popular cell line, although quite restrictive in terms of the ability, its ability to be used clinically. And finally, induced pluripotent stem cells, which is a source of cells from stem cells that get differentiated into mature NK cells and then used in immunotherapy. There are robust procedures right now to differentiate NK cells from induced pluripotent stem cells, and they are being used to develop these off-the-shelf therapeutics wherein NK cell or engineered NK cell can be used as effectors in immunotherapy. When we talk about peripheral blood, the attractive thing about NK cell peripheral blood use is because they are, again, always on, and they operate under MHC mismatch conditions. So it makes them safer as an autologous or allogeneic source. Peripheral blood is a safer source of NK cells than it is usually of T cells. However, all of these cells, when they're sourced from these environments, these are really are sourcing healthy NK cells. So peripheral blood, cord blood, induced pluripotent stem cells give rise to very healthy cells from healthy donors, and not oftentimes cells from patients or cells that are in tumor microenvironments are much more dysfunctional than these ideal, so to speak, sources of NK cells. In order to enhance the specificity of NK cells in cancer, we build these cells through genetic tools, which allows us to engineer them with higher specificity for cancer recognitions. For example, chimeric antigen receptors, which are synthetic genetic receptors used to enhance specificity of immune cells to our target, are oftentimes engineered on the surface of NK cells to enable their recognition and even more specific elimination of target pathogens. Of course, CARs have been popular because we have 6 FDA-approved CAR T products in the market. They all target various blood malignancies. CAR NK cells are in clinical trials, and though we don't have an FDA-approved product, we have demonstrated that engineering NK cells with chimeric antigen receptor can enhance their specificity multifold and enable them to act under even more immunosuppressive conditions to recognize cancer pathogens. Chimeric antigen receptors, of course, for those of you who don't know what they are synthetic engineered molecules. These are genes that get translated into proteins that are ultimately expressed on the surface of immune, such as T or NK cells, and allows these cells to recognize specific proteins or antigens on target cells and deliver an activation signal to the cell. This activation signal comes from the transmembrane intracellular domains of CARs. These are also called co-stimulatory domains, and there's multiple types of them. 3 are listed in this slide, such as 4-1BB, CD28, and C8. CARs are usually designed to recognize either tumor-specific or tumor-associated antigens. Most of the CARs, including all the FDA-approved CAR T-cell products, are designed to target tumor-associated antigens. These are antigens that are overexpressed in tumors, but are not entirely absent from healthy tissue. On the other hand, tumor-specific antigens are antigens that are almost specific to the tumor. These are the ideal antigens that really avoid off-target effects because they're almost absent from healthy tissues. Unfortunately, identification and targeting through genetic tools or CARs or other target therapies of tumor-associated antigens is very difficult because it's really it's tough to identify tumor-specific antigens for a lot of cancers. While a lot of our initial CARs for NK cell therapy, NK cell engineering, have been made and designed using T-cell architecture constructs, we have recently started developing specific genetic tools that are utilizing signaling domains that are specific to NK cells. So for a long time, we took CARs that had been used for T cells and put them straight into NK cells. However, recently we really learned there are specific transmembrane and stimulatory domains that can be engineered into a CAR that are specific for NK cells. For example, CD3, beta, DAP10, NKG2D are specific of Fc gamma are specific for NK cells and may work better in CAR architecture. Ongoing studies are evaluating the ability to use these NK-specific CARs to enhance the ability of NK cells to be genetically engineered for cancer immunotherapy. The workflow for an NK cell therapy is illustrated in this slide. We talked about NK cell sources. NK cells can, of course, be sourced from peripheral blood, NK cell lines, cord blood, or HSCs, iPSCs, and you can differentiate into mature NK cells. Then they can be genetically engineered either by adding a CAR or by knocking out receptors through genetic engineering or CRISPR. Once these cells are genetically engineered, they get expanded, frozen, and then the patient gets treated by adoptive transfer. In an autologous setting, the sources of NK cells would be the patient. In an allogeneic setting, the source of NK cell could be any of those 4 sources listed on the first slide. And of course, allogeneic therapies allow manufacturing to be maybe even quicker, but definitely easier, as it does not require invasive procedures where the patient would have to donate blood to make cells. And for those of you more familiar with CAR T cells, there are a lot of similarities between CAR T and CAR NK cells. CAR T cells are, of course, can be sourced also from peripheral blood, cord blood, and iPSCs. CAR NK cells have also all of the sources, including iPSC, which are methods of which are probably more robust for NK cells than T cells. The main difference between CAR T and CAR NK cells are their ability to T cells' ability to last for a long time. So they are long-lived cells. On the other hand, CAR NK cells without any kind of support have very limited persistence. One of the main considerations for CAR NK over CAR T cells is the safety. CAR T cells are usually associated with cytokine storm and the potential for GvHD in certain settings. On the other hand, CAR NK cells are safe. They are not considered to be involved in the induction of GvHD or cytokine storm, meaning that there's no CRS risks. In terms of engineering transfection efficiencies that they're used, they can be used similarly. The difference being that CAR NK cells can be used allogeneically, a huge advantage for manufacturing of cell therapies, whereas CAR T cells have to be used with MHC match. So you need to have completely matched donor to recipient to be able to use these cells efficiently in therapy. CAR T cells, of course, have been successful because we have 6 FDA products on the market. However, all those products target various blood malignancies. This is data that is now a couple of years old, 2 to 3 years old, but the trend remains much the same. The x-axis lists a number of antigens that are being targeted through CAR T therapy, and the color coding represents gray for no response or disease progression. The peach color is stable disease, and the green is complete response or partial response. We, of course, want as much green as possible, or at least peach means the patients are stable. However, for solid cancers, most of the patients don't respond very well. There are some cancers that are more responsive than others, but for the large part, most of these antigens have resulted in more non-responding patients than responding patients. And this is a problem. The problem is that for a lot of CAR T or CAR NK therapy, solid tumors represent a true obstacle to the efficacy of these therapies. And that is, again, why we don't yet have a solid tumor CAR T approvals or CAR NK approvals, but we have 6 FDA approvals for blood cancers. And of course, a lot of studies have been done to try and understand why solid tumors have been so difficult to target with CAR T therapy. And so various types of transfection methods have been tested: longer or shorter cell duration, cell expansion duration, longer or shorter persistence, transfection efficiency, dosing, and whether you add cytokine to support and stimulate CAR T cells further in vivo. However, for all of these methods in solid tumors, and this is data from solid tumors, the pool response rate has remained less than 20%, meaning patients didn't respond no matter how you prepare these cells. There are many answers for why this could occur. So for to understand some of these answers, we looked at data from brain cancer, so glioblastoma patients. This is TCGA gene transcription data that we extracted. And we looked at a number of genes that correspond to favorable conditions or unfavorable conditions in the tumor environment. We identified NK cells through 5 genes, and we correlated them through proteins or genes in cancer that represent inhibitory conditions and some that represent activating conditions. For inhibitory conditions, we use CD73, for example, CD155, GD2. For activating conditions, we use the NKG2D ligands, MICA, MICB, IL-15 a cytokine, and CXCL10, CCL5, 2 chemokines that are known to recruit NK cells. And so we found that NK cells are actively excluded from environments of the tumor that have a lot of inhibitory functions. So active exclusion of immune cells from environments that are immunosuppressive has been really is really a mechanism that tumors use to exclude immune cells. However, when those tumor patients that a lot of favorable ligands, so IL-15, CXCL10, CXCL5, NKG2A ligands, the NK cell had a higher percentage of presence in those patients. And so the correlation was that NK cells are actively attracted to environments where they can survive in, which of course makes sense, but this is what our brain cancer patients also seem to favor. There are, of course, many reasons why cells are excluded from environments, and the previous data really just showed a snapshot of those. The NK cells, especially in cancer, for many reasons of heterogeneity, antigen escape, and conditions of hypoxia, low pH. There's also active metabolites and oncometabolites, and things such as adenosine, lactate, MTA, TGF-beta. Inhibitory ligand checkpoint inhibitors, PD-1, TIM-3, TIGIT, many of them are upregulated, which cause a dysfunction of immune cells. And of course, poor cell infiltration because of dysregulated chemokine receptor ligand axes. The tumor stroma is very impenetrable, and also an unfavorable ECM, and also the vasculature of the tumor, which is impenetrable and which creates an impermissive environment. So there are many reasons why NK cells are infectious in cancer. We do know that a lot of those reasons have been studied through immunotherapeutic approaches, including the targeting of checkpoint receptors. However, for a lot of tumors, a lot of patients, we don't know what receptors to target because we don't know what receptors are upregulated and what receptors would provide the best type of response. We do know from our work and that of others that NK cells have heterogeneous responses in cancer, and so some receptors that are upregulated dysfunctional in T cells do not act the same in NK cells. And so context-specific activation is required for NK cells. We do know, for example, that NK cells that are in patients that are not equally metabolically active, so metabolic fitness, so for example, CD56 dim NK cells, allow them to have a higher glycolysis rate while CD56 bright NK cells, which are the most, which are considered a less mature type of NK cell, make a lot more cytokine. We can use cytokines not only to enhance NK cell life in vivo. Patients given NK cell therapy need to receive cytokines for this, or need to have some kind of support for NK cells to survive longer than a week. And so here, this slide lists various cytokine regimens that are used to activate NK cells. Some of them give rise to memory-like, such as IL-12, 15, 18. Some will give rise to higher effector responses to IL-12, 15, without any change in metabolism. One of the receptors that are actually involved in regulation of metabolic activity of NK cells is TIM3. We know that TIM3 has higher IFN gamma and GBM, and we also know that TIM3 expression in NK cells changes with glucose availability. We do, however, also know TIM3, which is a dysfunctional receptor on T cells, is not dysfunctional in NK cells. And so we have this difference in responses. And this is an example of how we cannot always really translate T cell immunology to NK cell immunology, because dysfunction of T and NK cells is different in cancer. One of the targets that's actively involved in immunosuppression of both T or NK cells is adenosine. Adenosine is made by an enzyme called CD73, which is upregulated in a lot of cancers. Adenosine is produced from ATP metabolized by cancer through CD39. CD39 then takes adenosine and converts it to AMP. Takes ATP and converts it to AMP, and then CD30 takes AMP as a substrate and converts to adenosine. Adenosine then binds to adenosine A2I receptors on NK cells and shuts down their function. CD73 is a prognostic factor in a lot of cancers, including lung cancer, glioblastoma, and many other cancers. It is also a prognostic factor in lung cancer, which is some of the work that we've done on CD73 has been in lung cancer. And also we've studied various patient-derived tissues to show that CD73 is very, very heavily expressed. We do also know, and this is all published data, so I won't go into too much detail, that adenosine causes downregulation of metabolic control genes and also a lower cytotoxicity of NK cells that are stimulated with IL-2 and IL-15, and this cytotoxicity would only be restored if its metabolism was impaired using adenosine deaminase. It was proven that it's inhibitory once adenosine deaminase was used. In order to counteract the inhibitory activity of adenosine, one of our very first studies was to combine CD73 antibody blockade with NKG2D engineering of CAR NK cells. So we used an NKG2D CAR fused to a DAP10 signaling domain, and then we transduced NK cells with it, and we combined it with the antibody blockade of CD73 using a antibody that is able to block the enzymatic activity of CD73, so it did not produce a CD73 anymore. The effect was modest, but it allowed us to see that trying to eliminate adenosine from the tumor microenvironment was beneficial. These treatments also resulted in higher infiltration of NK cells in the tumor, but also they allowed us to see that it was beneficial to eliminate CD73 from tumor microenvironment. As a second step, then we moved past antibody blockade and we developed a CAR that directly targets CD73 via Fc16 ligand domains. And again, this is also published data. And we showed that peripheral blood NK cells can be transduced with this CAR, and they can be very effective at eliminating cancer. They not only prolong survival of adoptively transferred lung cancer xenografts, but also they showed really strong anti-tumor efficacy. These cells also had high granzyme production in the tumor, homed more potently, and also acted very functionally comparatively to healthy blood-derived NK cells. The problem with that is that when we use healthy NK cells, such as the one that we've seen in the previous study, to engineer CARs with, we really are relying on very functional and active NK cells. Unfortunately, the tumors, NK cells are not very active. And so what you see here is GBM patients have been phenotyped. We phenotyped a bunch of over a dozen GBM patient glioblastoma for what we consider checkpoint inhibitors, and we see a very high differences between NK cell checkpoints in healthy tissue and in brain cancer patients. A lot of things don't agree with what we see in T cells, but we see higher PD-1 upregulation, higher LAG-3 upregulation, for instance, of these tumors, of these cells inside these tumors. This also tells us that NK cells are very specific, and that tools to really develop NK cell therapies have to be developed specifically for these cells, as opposed to taking too many cues from T-cell immunology. One of these receptors that is very highly upregulated in T cells that are dysfunctional is TIGIT. We do know that TIGIT interacts in a very wide range of networks. It's involved in a lot of in a wide network of many binding partners, and TIGIT is really most inhibitory and is bound to CD155, which is an antigen expressed in a lot of cancers, including particularly on solid tumors. We sorted NK cells for their expression of TIGIT, and we found that TIGIT-positive NK cells were actually more active than TIGIT-negative NK cells. We believe TIGIT is inactivating. TIGIT gets expressed as a part of function of activation of NK cells. It could lead to exhaustion after chronic stimulation. However, TIGIT-positive NK cells are not exhausted. TIGIT-positive NK cells are able to make more CD107, more alpha and gamma, have higher lactate concentration, sort of higher metabolic activity, and also they're able to kill targets more than TIGIT-negative NK cells. They also have multiple pathways of activation when we did RNA-seq of these cells to show that TIGIT is not an inhibitory receptor for NK cells unless it interacts with CD155. However, a lot of the times tumors have mechanisms beyond upregulation of inhibitory receptors to fight off immune cells. One of these involves the expression or underexpression or overexpression of chemokines and chemokine receptors. Immune cells traffic to tumors in response to a lot of chemokine receptors and ligands. Some of these chemokine receptors are suppressive to the immune system because they actively exclude immune cells, effector immune cells, by recruiting immunosuppressive cell populations. Some are activating. However, in patients, as this graph at the bottom shows, chemokine receptor axis is dysregulated, while CXCR3 is a really important ligand for NK cell migration. In tumors, particularly in patients, these are dysregulated, expressed in aberrant ways. And so while we are looking for high expression of CXCR3, patient donor GBM patients have dysregulated levels of CXCR3 in their blood and in their tumors. We do know that CXCL10, for example, is a very active immune attracting chemokine, and we found that in GBM tumors, it is very important to have higher levels of CXCL10. As we can see, NK cells are very actively recruited to tumors that have higher levels of CXCL10. In order to combine the ability to traffic and to target some of these mechanisms of immunosuppression, we developed a triple CAR NK cell that uses NKG2D and GD2 as their antigen, so cancer cell recognition ligands. And CD73 was cleaved via a peptide linker, cleaved by proteases in the tumor microenvironment. A three-dimensional version of this CAR is illustrated in this slide. It shows that our CAR NK cell has three components: an NKGD in depth 10 CAR that signals via CD3 zeta, a GD2 CAR with a CD8 hinge, CD28 and CD3 zeta signaling domains, and a CD73 antibody fragment that is cleaved via tumor-associated proteases, and then it gets released to block the production of adenosine via interaction with CD73 on the cancer cell. Of course, we used a lot of assays to show that we can take peripheral blood NK cells and then transduce them effectively. And this is, again, published data that is easy to look up to see that human NK cells can be effectively transduced with these genes. And we also combined this treatment with with a way to recruit NK cells to the tumor. So we found that if we treat brain cancer tumors with chloroquine, we found that these tumors release a lot of CCL5 and CXCL in particular. Both of these chemokines are very attracting NK cells. And so when we combine chloroquine inhibition of brain cancers, these brain cancers start producing high levels of CXCL10 and CCL5, which in turn recruit NK cells. By then adoptively transfer CAR NK cells, we get a higher concentration of local NK cells into the tumor. This was confirmed via our xenograft studies, where we implanted intracranially, so orthotopic brain cancer brain tumors into mice, and we treated them with adoptively transferred triple CAR NK cells together with chloroquine. When the combination therapy was delivered, these mice had really remarkable anti-tumor responses, both in terms of survival and in terms of anti-tumor effect. And then we found that not only was chloroquine effective in removing adenosine from the tumor, but also the chemokines released by these brain cancer cells effectively recruited NK cells because of their high levels of production of CCL5 and CXCL10. We did a lot of studies both in vivo and in vitro that NK cells are able to cross the blood-brain barrier, which would be a next strategy to deliver these cells, perhaps IV, and see whether they can migrate into the tumor. Our previous study, the NK cells were delivered locally into the tumor intracranially, as that was the most effective way to establish efficacy of therapy. But as I mentioned, I think that there's more work to be done in our ability to see whether we can actually deliver NK cell into the brain in a way that can hopefully recruit higher numbers of NK cells across the BBB, and if that's a therapeutic opportunity. And for the last part of the talk, I really want to highlight some efforts into developing off-the-shelf NK cell therapies. iPSCs are attractive because they're able to enable not only allogeneic therapy, but they're renewable, donor-independent, and less invasive, as they don't require donors patients to donate their own blood for NK cell therapy. They also can be genetically manipulated a lot easier than NK cells, which is one of the biggest challenges to NK cell. However, challenges include the long protocols that are required to differentiate iPSCs into NK cells. The purity and the immunology of these systems have not been fully studied, so we have a lot of open questions about what kind of cells we are able to produce. A little while ago, we developed a protocol that enabled us to produce NK cells from iPSCs over a period of about 40-45 days from stem cell precursors. We showed that we can use centrally validated iPSC cell lines and differentiate them into mature NK cells. These cells are very pure, they're able to kill, and the maturation and differentiation really follows cytokine-driven differentiation of NK cells. By doing that, we try to recapitulate the natural process of NK cell development in vivo. The cytokines that are used, as CF, IL-12, 15, IL-12, IL-18, are some important cytokines in NK cell development, and they all functionalize NK cells in different ways. So over the period of these 3, 4, 5 weeks, we really give NK cells cytokine-driven stimuli that enable us to differentiate and get to NK cells that are as much alike peripheral blood mature active effector NK cells as possible. Of course, these cells are pure. They express all of the receptors. Again, this is all published as regular NK cells, and they also are able to be expanded to clinical doses. So we can make NK cells from iPSCs and expand them sufficiently to be able to have the kind of purity and levels needed for adoptive transfer therapy. And really, this is one of our approaches in engineering iPSC NK cells into supercharged engineered NK cells for solid tumors. Here we're using a SynNotch system that utilizes TIGIT and CD155 as ligands on cancer. So the construct is based on TIGIT interaction with CD155 on cancer cells. When TIGIT expressed by the SynNotch synthetic receptor interacts, it releases a transcription factor, which triggers the translation of CD73 antibody fragment, which gets released from the cell and finds CD73 on cancer and blocks it. So this SynNotch is a gate-based activation. If the NK cell binds TIGIT, CD73 is released. We differentiated our iPSC NK cells with this construct and showed that they can not only express successfully the construct, they can also carry through the differentiation process and result in mature NK cells. We then treated our NK cells with these adoptively transferred SynNotch engineered constructs and find that have really remarkable anti-tumor effects. Not only are the tumor effects remarkable, but we found not only survival and tumor growth, but also almost elimination from the tumor for multiple mice treated with this adoptively transferred SynNotch iPSC NK cells delivered locally into the brain. So this allowed us to show that we can not only use stem cells as a way to make NK cells and engineer them, but also use them potentially in a really remarkable and powerful way against very aggressive tumors. Again, these cells were again injected intracranially into the brain. This is a local treatment, but a good first step to show efficacy of iPSC-derived NK cells. There are considerations and lessons that we've learned through engineering iPSC NK cells, but it's a first step towards hopefully having an allogeneic off-the-shelf therapy with a cell line that has effector capability, so NK cells, but it's also synthetically generated. With this, I've reached the end of my talk. I want to thank everybody for your attention and for listening. And I really want to summarize, so hopefully I gave you a little bit of flavor about the power that NK cells have in immunotherapy, how we need to be smart about understanding specifics about NK cells that are different from maybe other cells, such as T cells, and how we can use that knowledge to develop really strong and powerful and potent immunotherapies through genetic engineering, synthetic biology, and even combination therapies, recruit NK cells to the tumor, but also act on multiple elements of immunosuppression at the same time. So with that, I want to thank everybody. Thank you, and thank you for listening.